Welcome to Learning Science is Fun. I am Sarah Manuel, and today we will learn about cardiac troponin I. Troponin is an intracellular protein that plays an important role in regulating striated muscle contraction. It is a calcium regulatory protein for the calcium regulation of contractile function in skeletal and cardiac muscles. Troponin is first discovered in 1965 and was originally named tropomyosin-like protein because troponin and tropomyosin are both regulatory proteins. However, in 1973, it was purified and characterized from skeletal muscles of a rabbit as a separate complex consisting of 3 protein subunits and later on termed as troponin. Troponin is a heterotrimetric protein complex which is arranged in a 1:1:1 stoichiometric ratio along the thin filament. The troponin T is the largest of the 3 subunits. It secures the troponin complex to the myofibrillar thin filaments via its interaction with tropomyosin. Next is the troponin I, which is the inhibitory subunit that prevents actin-myosin interaction during muscle relaxation. Lastly is the troponin C, which is the smallest subunit that binds calcium ions to initiate a muscle contraction. When we look at the genetic level, there are 8 distinctive troponin genes that code for tissue-specific, heart, and skeletal muscle protein subunits. These genes are isoforms, meaning they have similar but not identical amino acid sequence. Troponin I has 3 isoforms encoded by 3 homologous genes. As you can see, unlike troponin I and troponin T, troponin C does not have a unique cardiac isoform. The tight regulation of troponin, specifically cardiac troponin, at both the transcriptional and translational level play an important role in the cardiac development, health, and muscle pathology. Cardiac troponin I is a muscle protein with a molecular weight of 24 kilodaltons and consists of 210 amino acid sequences. Troponin I differ from other troponins due to its N-terminal extension of position 26 amino acid. As you can see from the screen, the highlighted Y is the amino 26, and on the left you can see the extension. This extension contains 2 serines in position 23 and 24. Presented on the screen is the ribbon diagram of human cardiac troponin core complex and its different subunits. The blue parts represent the troponin C, the red parts represent troponin I, and the green parts represent troponin T. Catalytic and structural calcium ion are shown as gray spears. Also, the inhibitory peptide and the switch peptide of troponin are also indicated. On the screen is the chromosome which contains human cardiac troponin I. By somatic cell hybrid analysis, the locus for cardiac troponin I gene, TNNI3, can be localized to the region 19q13.42. This means that the exact location is at chromosome 19, on Q or the long arm, band 13, and subband 42. The subunits of troponin works cohesively to regulate the contraction and relaxation activities of striated muscles. The troponin C binds calcium ions, the troponin T interacts with tropomyosin and anchors troponin to the actin, and the troponin I inhibits the ATPase activity of the actomyosin cross bridge and effectively blocks the myosin binding site on actin subunits. When cardiomyocytes are damaged, the cystolytic pole is released and then troponin is also released into the blood circulation. Elevated levels of troponin indicates myocardial damage. The normal level of troponin I in the body is less than 0.035 nanograms per milliliters. After a myocardial infarction, troponin I is released into the bloodstream, making the level of troponin I to spike. It will take about 3 to 4 hours before the level elevates, and it will peak at 18 to 24 hours after the myocardial infarction. The troponin level will return to its normal itself after 1 to 2 weeks. This level is used to measure and evaluate the damage to the heart muscles and diagnose myocardial damage after a myocardial infarction or cardiac arrest. Over 100 mutations in cardiac troponin subunits have been linked to cardiomyopathy or disease of the heart. This is because a change as small as 1 amino acid in the troponin sequence can result to a mutation and cause changes to troponin subunits interaction in the actin-myosin cross-ridge formation, muscle contraction, and cardiac function. When a patient is having a heart attack or myocardial infarction, the level of troponin rises, which can also result in unstable angina, which is a dangerous type of chest pain. When a patient had a heart attack, it can result to a serious disorder like acute coronary syndrome, heart failure, pericardial disease, and cardiomyopathy, just to name a few. The National Institute of Health and National Heart, Lung, and Blood Institute define cardiomyopathies as a group of diseases of the heart that are either inherited or acquired. It is discovered that a variety of hereditary cardiomyopathies have been associated with mutation in the three subunits of the cardiac troponin. Some of these hereditary cardiomyopathies are hypertrophic cardiomyopathy, or the thickening of the heart muscle, restrictive cardiomyopathy, or the stiffness of the heart muscle, and dilated cardiomyopathy, or the thinning of the heart muscle wall. I guess that ends our discussion. Thank you so much for listening, and I hope you learned something. Here are the lists of my references if you want to read and learn more. To learn more about other proteins and their functions, check out our website at bsedsciencepup.com, and always remember, science can never be hard because learning science is fun.